Assalamu alaikum jami'an. So today we will start chapter 4 about the chromosome theory of inheritance. So as you can see here in this image, the structure of the chromosomes. So Mendel studied inheritance without being able to see those subnuclear structures and without being able to follow exactly what happens during meiosis. So in this chapter, we will know that our genes, the different alleles are carried on those nuclear structures, the chromosomes, and we will see the behavior of these chromosomes during the two types of cell divisions, the mitosis and the meiosis, and we will end by concluding that indeed genes are residing on the chromosomes based on certain observations. So similar to mutations in specific genes, what we call monogenic mutations, we can get abnormalities by inheriting abnormal number of a chromosome or structural abnormalities of chromosomes. So sometimes when individuals they get extra chromosome 21, they will have mental abnormalities or learning disabilities. A syndrome is known as Down syndrome or trisomy 21. Similarly, individuals with less chromosome, with one chromosome less than normal, they will have also certain abnormalities. So genetic problems can result because of mutations in single genes or because of also chromosomal abnormalities. And the science that deals with chromosomal numerical abnormalities or structural abnormalities is known as cytogenetics. So we will talk a little bit history in which we will go before Mendel, 200 years before Mendel, in which Leeuwenhoek, an astute microscopist, was able to develop uh, microscopes, and using this microscope he was looking at semen, and he was able to see the sperm cells in the semen, and he hypothesized that during fertilization there will be a meeting between an egg and the sperm. So after that, but this was just a hypothesis, so he was not able to follow actually the fertilization process. 200 years after Leeuwenhoek, uh, scientists they were able to study fertilization in organisms with the large egg like the frog and the sea urchin, and they were able to see that during fertilization actually a sperm and the egg fuse together. So they concluded that the nuclei uh, in those cells they fuse together. Uh, then by further improvement with the microscopy and also the development of a specific stain by Walter Fleming, um, he was able to stain specifically chromosomes inside the nucleus, and after that time chromosomes were named chromosomes representing or you know denoting for the bright bodies. Okay, and by following the behavior of these colorful bodies or bright bodies during the different cell divisions like mitosis and meiosis, they concluded that the behavior of these chromosomes they resemble the behavior of alleles that Mendel described, which suggests that our genes are residing on these chromosomes, or chromosomes are the carriers of our genes. So if we take diploid cells or somatic cells from Drosophila melanogaster, both males and females they will have 8 number of chromosomes. So remember that diploid number we denote by 2n. So here 2n equal 8. If we are talking about a human, 2n is, or the diploid number is equal to 46 chromosomes. So these are the diploid cells, the somatic cells with full complement of the chromosomes. And if we look at the sexual cells or the gametes, we can see that gametes, they have only half number of the chromosomes. That's why they are called the haploid cells. Haplo means half. Okay, so the haploid number of the chromosomes in Drosophila is 4. The haploid number of chromosomes in a human is 23 chromosomes. So during fertilization, when the two haploid cells, the sperms and the oocyte, or the pollen and the ovules, they meet, they will maintain back the diploidy in the zygote, and then the zygote will start to divide mitotically. So mitosis maintain diploidy, while as we will see that meiosis reduces or will produce half number of chromosomes in the gametes. So if we look at the general structure of these chromosomes, so chromosomes, they will vary in length, and also they will vary in the location of the centromere. The centromere is the maximum constriction of the chromosome, or also called the waist of the chromosome. So sometimes the centromere location is central, and these kinds of chromosomes are called metacentric because the centromere is at the middle. Other types of chromosomes, the centromere are near or close to the end of the chromosomes in the acrocentric chromosomes. So chromosomes are divided to metacentric, acrocentric. Also, we have submetacentric when the chromosomes are off center, a little bit far from the center. They are not at the middle, but they are also not close to the end. They are called submetacentric. Uh, and in certain organisms and in abnormalities, uh, sometimes the centromere will locate it just at the tip of the chromosomes, and it's called telocentric chromosomes. So again, to analyze the anatomy of a chromosome. So basically what we see here, we see pairs of homologous chromosomes. So in any diploid cells, we will find that each chromosome has a chromosome that looks very similar to it, at least when we look at with the microscope, or when we analyze it with different staining techniques. So each chromosome has another chromosome very similar to it. It's called the homologous pair. So we divide the chromosomes in diploid cell in pairs. So and these chromosomes, they will have same centromere location. So if one is metacentric, the other one is metacentric. Um, they will also have the same length, and they will also have the same uh, arrangement of genes. So if we find here the gene for the isoglutanin, let's say, the other homolog will have another copy of the isoglutanin, either the same allele or a different allele, but the sequence of the nucleotides in the same location, it's called the locus. So the location of a certain gene on a chromosome is called locus, and the plural is loci. So these homologous chromosomes, they will have identical location or locus of different genes, of the same genes, but maybe a variation of the gene copy or the alleles. Here we can see these chromosomes. This pair of a chromosome has also uh, acrocentric chromosomes, so the centromere is toward the end of the chromosome. Uh, so they will have also different kinds of genes, but the arrangement of genes in both of these homologous chromosomes, they will be the same. So homologous chromosome, they share the centromere location, uh, the same length, and also the arrangement, the same arrangement of genes. What help us to identify as cytogeneticists, to identify these homologous chromosome in any cell is to do a staining technique uh, like G banding, Giemsa staining, or C banding, different kinds of banding techniques in which the chromosome will appear as dark band and light band. Dark bands, they will absorb more of the stain, while the lighter band, they will absorb less. And the reason why the dark band absorb more is because it has a heterochromatin, a lot of uh, uh, folded chromatin, and it represents usually inert uh, genes or in inert part of the chromosome in which the genes are not expressed. So what remains to say here is that in each chromosome, so these are pair زوج من الchromosomes, and in each chromosome we will find two identical sister chromatids. So replicated chromosomes, when they finish S phase, each chromosomes will be represented by two identical copies, and these are called the sister chromatids. And from where the centromere is located, we will see also arms of the chromosomes. So these are two arms and these are two arms. So chromosome will have a replicated chromosome will have four arms and two sister chromatids. So what is really helpful to identify a numerical or structural abnormality is to do a karyotype. So you take a sample, a cell sample, either blood sample or from the bone marrow or from a cancerous tissue, depends on the reason to take the sample and the reason why you need to do the karyotype analysis. So karyotyping is actually, or a karyotype is the graph that shows an arranged chromosomes, and the chromosomes will appear as copies, two copies of each chromosome. So in a human, we have 23 pairs of chromosomes. And as you can see here, the chromosome will appear like a dark band and light bands. And these are the light bands are the euchromatin and the dark ones are the heterochromatin. So all of these pairs of chromosomes, they share, as we said, the same length, the centromere location, and the same banding patterns. What we cannot see here is actually the genes, the location of the genes, and the type of genes. Uh, so all of these 22 pairs of chromosomes are called autosomes because they do not determine sex. So genes that are carried on these pairs of chromosomes, uh, uh, they are important for different phenotypes. They produce enzymes, they produce different structural proteins, but they are not needed, or they are not determining the sex, while the last pair here is called the sex chromosomes. And in males, we have two different ones, the X and the Y. So these are non-homologous chromosomes because they have different length, they have different centromere location, and they also have different arrangement of genes. So sex chromosomes. So Walter Sutton was one of the first scientists to correlate between the inheritance of uh, specific chromosome and sex determination. So this scientist was interested to understand how sex is determined, and he followed the chromosome segregation in grasshoppers. So grasshoppers' diploid number is 24 chromosomes. Uh, so they have 22 matched pairs, 11 pairs of autosomes, and one pair of X and Y, similar to a human. And he followed meiosis in males and in females. So females, they will have 11 autosomes and 2 X chromosomes. So he followed spermatogenesis and oogenesis, and he found that sperms with the Y chromosome, when they fuse with an egg, they will form males, while sperms with X chromosome, when fused with the oocyte, they form a female. So he concluded that the presence of these sex chromosomes determines sex, and this was the first phenotype that is linked to a chromosome. So sex determination, uh, uh, discovered by Walter Sawton in grasshoppers, is the first phenotype that is linked to uh, chromosomal inheritance. So as I just said, fertilization of an egg with a sperm will form two X's plus the other 12 pairs of autosomes, and this would result in a female, while two sex chromosome X and Y, they will form a male. So he concluded that it must be something present on these sex chromosomes that determine males and females. Similarly, in a human, sex determination depends on the sex chromosomes, in which males they are XX XX karyotype females, while males are XY. Uh, and you can see here the difference in size between this is the Y chromosome and this is the X chromosome, and you can see that X chromosome is submetacentric, while Y chromosome is acrocentric. So even the location of the centromere is different between the X and the Y. Uh, so here, males, they will produce two kinds of gametes, 22 chromosome and X, or 22 chromosome with a Y. So uh, while the females, they will produce 22 uh, they will produce eggs with a 22 autosome and X chromosome. So all eggs, they will have X chromosome. That's why we call males as heterogametic. Heterogametic means that uh, produce two different heterogametic. I see. While females are homogametic. So males can produce sperms with two different um, X chromosomes with either X or Y, while all the eggs, they will have the same kind of X chromosome. Uh, what was really important, so we know from the presence of Y in either a grasshopper or in a human, and that is linked to maleness. So if you inherit Y chromosome, you will become a male. Then it must be that there is something on the Y chromosome, a gene on the Y chromosome that determine maleness. And they discovered actually a gene. It's called the sex determining region on the Y, the SRY gene, located on the tip or in the short um, arm of the Y chromosome. And the way how they discovered that is by studying the sex reversal individual. So normally XX, uh, they are females, while XY are males because of the presence of the SRY gene on the short tip of the Y chromosome. So what happens sometimes, and this is very rare, you get a karyotype of XX, but then when you look at the phenotype, it's a completely normal male. So a male with a testicles and a penis, while the karyotype is XX. And the reason of that is that the X chromosome now has SRY gene. So if SRY gene is translocated, so a kind of interchange or a mix or a movement of SRY from the Y chromosome to the X chromosome, then if we have X chromosome with SRY gene, this will result in a male, and we call it a sex reversed male because the karyotype and the phenotype is not um, normal. So we expect that XX produce a female, while here it produces a male. Similarly, if we have XY karyotype, but with either a mutation or a deletion, so any kind of a mutation in the SRY gene, this will result in a female phenotype. All right. So these sex reversal individuals, especially those XY females, they will be infertile because the remaining Y chromosome has genes. Uh, I mean here the XX females here, they will be phenotypically male but infertile male because uh, the remaining Y chromosome, on the long arm of the Y chromosome, there are genes that are important for fertility. So sex determination in a human depend on the presence of the Y chromosome. So the absence of Y chromosome will make individual female. In Drosophila, it depends on the number of X chromosome. So if the individual have one X, it will produce males, while two X or more produces females. So let's look at the table to see the differences between Drosophila and human sex determination. So in Drosophila, having three X's, they die, while in a human they will look as a nearly normal female. XX is a normal karyotype in human and in Drosophila. So here is the distinction. XXY in human is a male with a Klinefelter syndrome, while in Drosophila it's a female. Remember that Y chromosome in Drosophila has nothing to do with sex determination, but still is important for fertility. So as a rule, because this individual has two X's, it will become a female in Drosophila. X1X, sometimes we write it as XO or just X, uh, is a sterile male in Drosophila because the absence of the Y chromosome, while in a human it's a female with a Turner syndrome. XY is a normal karyotype in both Drosophila and the human. XYY is a normal male in both Drosophila and the human, and the absence of the X chromosome will cause lethality. So this table is important to know the differences of sex determination in Drosophila and in human. Other organisms, they have uh, other kinds of sex determination. Uh, so we said that in human and in Drosophila, the males are homogametic. The males are, sorry, heterogametic because they produce two different kinds of gametes, while females are homogametic. In birds and butterflies, it's the opposite. Sex chromosomes are different, Z and W, but the females are heterogametic while males are homogametic. Bees and wasps, when fertilization happens, when diploid organisms are produced, all of these diploid organisms, they will develop into females, while the haploid ones will develop into males. In lizards, temperature affects sex determination. In certain kinds of fish, when they are young, there will be males. When they become older, they turn into females. So it's really amazing how different organisms, they have different mechanisms of sex determination. So as I said at the beginning of the chapter, so after discovering of those chromosomes, the bright structures in the nucleus, they followed the behavior of chromosomes in mitosis and in meiosis, and this gave them an indication that the behavior of these chromosomes, the segregation pattern of these chromosomes, is very similar to the behavior of alleles that Mendel described. So let's go through mitosis, the nuclear division that occurs in somatic cells, and then we will talk about meiosis, the reduction division that occurs in the germ cells. Uh, so remember that the cell, dividing cell, it has to go through sequential stages. It's called the cell cycle, in which it's composed of G1, the gap phase or growth phase 1, the cell grow, produce proteins, increase the number of organelles, and also at this stage they are waiting to receive signals in which to tell the cell to go to the next stage. And then in the next stage is the S phase, the DNA synthesis stage, in which the DNA is replicated and the chromosomes will be formed from two sister chromatids. Then another short uh, gap 2 stage, and then the cell enter mitosis. Certain structures within the cells are important during mitosis. These are called the centrosomes. So centrosomes are microtubule organizing center. From the name, they organize the formation of the spindle fiber. And the spindle fiber is composed from microtubules. So the main function of centrosomes in animal cells is to organize the formation of the microtubules. And these centrosomes, they are composed from two centrioles. So replicated cells, or cells that they are about to enter mitosis, they will have two centrosomes, and in each one there is two centrioles. This is the different stages of the cell cycle. G1, the gap phase, and non-dividing cells, they exit the G1 and they will enter a stationary phase, the G0. S phase chromosome duplication, short G2, and then uh, nuclear division mitosis, and in most kind of cells this is accompanied by split of the cytoplasm that is called cytokinesis. And what happens is that if we have a diploid cell with 2n equal 4 in this example, and we have here a pair of homologue, and we can see here each one will have two different alleles because these are heterozygous in this example, and another homologous pair of chromosome with also heterozygous allele of B. Then what happens during the S phase or after the S phase is that the chromosome number remain the same. So here still we have 4 chromosome, 2n equal 4. But what happens actually after the S phase is that each chromosome is composed from two sister chromatids. So the amount of DNA is duplicated, but the number of chromosomes remain the same. So these are the 5 substages of mitosis. We will go through them quickly to describe the differences in each stage. So here, and it's very important to focus on the centrosomes, the nuclear envelope, and to the chromosomes themselves. So we can see here that at the beginning of mitosis, what is called early prophase, the centrosomes will be near the nucleus, and the two centrosomes are near each other. And we can see here microtubules radiating from those centrosomes. At the beginning of this stage, the prophase, we will see that the nuclear envelope, the membrane surrounding the nucleus, And the chromosomes at this stage, they start to condense, they become thick and short, uh, in which uh, we can we cannot see actually that each chromosome is composed from two sister chromatids. But what is important is that the centrosomes uh, will start to move away from each other, and while moving away from each other, they will start to form or to organize the formation of the spindle fibers. Prometaphase is an intermediate stage between prophase and metaphase, in which the centrosomes reach the poles, the opposite poles, and while they reach the opposite poles, we can see here that the nuclear envelope is gone or fragmented. And we can see here that some of these spindle fibers are attached to a special um, place in the chromosome called the kinetochore. So the kinetochore are actually complex of the proteins that are formed near the centromeres, and they help to connect spindle fiber with the chromosome itself. And then in the next stage, metaphase, from the name, all the chromosomes will be arranged at the metaphase plate at the middle between the two centrosomes, and each chromosome is connected to spindle fiber from the opposite pole. So if we have here a chromosome, and these are the two sister chromatids, so from one pole, this chromosome will be connected, and here of the kinetochore, and another kinetochore in the other pole, which is important to connect the chromosome to the opposite pole. So at this stage, up to this stage, what keeps these sister chromatids attached together is a protein called cohesin. So at the beginning of the anaphase, separase cleaves cohesin, and then these sister chromatids will be free to move, and this is accompanied by the shortening of the spindle fibers and also motor proteins that work on the spindle fiber. They help to move these, and now we cannot call them sister chromatids, we can call them daughter chromosomes to the opposite end. And then in telophase, we will start to see reformation of the nuclear envelope, depolymerization of the microtubules completely, and decondensation of the chromatin. So chromatin will appear long, and here, because sister chromatids separated, so each chromosome will be formed from a single chromatin. Usually at this stage, it's accompanied by split of the cytoplasm, what is called cytokinesis. And in animal cells, cytokinesis is, is done by the contractile ring of actin, while in plant cells, so in animal cells, we can see the formation of the cleavage furrow and invagination because of the formation of the contractile ring. While in plant cells, because they have the cell wall which is really rigid, so they cannot do uh, they cannot do cytokinesis in a way of formation of a cleavage furrow, but uh, it's uh, uh, it's um, formed by the formation of the cell plate, uh, which is basically the fusion of organelles that have cell wall components that were formed from in the ER and then modified in the Golgi apparatus. They will make uh, a future cell wall and the plasma membrane between those two daughter cells, in which the cytoplasm will be split between the two cells. So cytokinesis is different between plant cells and animal cells. And in certain organisms and even in human, in certain types of cells, uh, nuclear division or split of a chromosome is not accompanied by split of the cytoplasm, so we end up having multi-nuclei in the same cytoplasm, and in animal cells it's called syncytium or syncytial embryo. So in Drosophila, the first 13 rounds of mitosis, mitotic division after fertilization, occurs without cytokinesis, and the embryo will end up having around 4,000 nuclei in the same cytoplasm. Later on in development, uh, formation of membranes or split of these nuclei occurs, which is called cellularization, and in which each cell will contain a single nuclei. So formation of multinuclei on the cetium is very common in animal cells and the plant cells during either in certain or specific cells or in certain developmental stage. So remember the cell cycle, G1S, G2M. So there are certain checkpoints, proteins and enzymes within the cell, which check that uh, all requirements are met in each stage before the cell are allowed to move to the next stage. So in G1, uh, the checkpoint check for the efficient size. Uh, they wait to receive growth factor signals, and then the cells will move to the S phase. Also, before the entrance of G2, uh, there will be another checkpoint, and also within uh, mitosis itself there's a checkpoint called mitotic checkpoint, and at this stage the checkpoint check if all chromosomes are attached to spindle fibers from both poles, or what is called a bipolar binding or bipolar configuration. So if there's any abnormalities in which either one kinetochore is not occupied, like in this example, I mean this kinetochore is connected while this one is empty, the checkpoint will remain on to prevent the missegregation of a chromosome. Or if both kinetochores are connected to the same pole, same centrosome, also this will cause spindle assembly checkpoint to remain active. Alright, so next time we'll start with meiosis. We will talk about the reduction division that will produce the gametes. See you then. Dr.